I really don't have any announcements today. So with that, I'll just introduce our speaker. We have James Boyko, who is assistant professor in ecology and evolutionary biology.
Great. Thank you, Marcy, for the introduction. Yeah. So I'm going to talk to you today about correlated character evolution models, but more specifically about how we use them in my field of comparative biology. So I want to introduce sort of the data types that we use, the kinds of questions that we're answering, and then at the very end, I'll run through a collaboration where we're looking at antibiotic resistance evolution. So I want to make sure I get through everything. This is like a mix of technical details and hopefully a good overview. So, like I said, I'm a comparative biologist, and in the field of comparative biology, it means that we are studying multiple species. So this is usually the scale at which I am thinking about things. I'm thinking about lizards and turtles and snakes and birds and like all of these different groups of organisms at the species level. So I'm sure most of you know this, but all species are part of a large tree of life, right? We are more closely related to gorillas than we are to these lizards here, to the birds. And this relationship is depicted in a phylogeny, which is this graph type of thing that you see here at the bottom. So you can see nodes and their shared ancestry. And this phylogeny is really important for the methods that we use because this is where a lot of our information is going to come from. And so the subdiscipline, I guess you could say, comparative biology is really never on its own as a thing. It's usually the subdiscipline of phylogenetic, which is because of the phylogeny, comparative biology. So we're going to use these species relationships and then descriptions of the species themselves to do our inferences. So the types of questions that we answer are related to species evolution. This is the abstract description, but I'll give you some examples of what X and Y could be. So we might be interested, does X evolve faster than Y? Now this could be the species itself, right? So you could say, does a species of Anolis evolve faster than a species of gecko? It could be a phenotype, does body size evolve faster than limb length? Things like this. We might ask, does a particular trait evolve before another trait? So is there a precursor before the evolution? Does change in a particular state then depend on X? And does X correlate with biodiversity, so the amount of species? So let's walk through an example. Here I'm going to use oviparity and viviparity. So oviparity is egg laying and viviparity is live birth. So here at the bottom, maybe I'll use, yeah. So here at the bottom, towards the base of the tree, we have this egg-laying species. So that's represented by this little egg here. And then we have different emergences of live birth. Okay, so that's what's represented here. That at the base of the tree, we have the most recent common ancestor of all of the species that are in the phylogeny, and we get the emergence of these different live birth lineages. So one thing that we might be, so this would answer the question, did oviparity evolve before viviparity? We would somehow reconstruct what the common ancestor of everything had, and then we would look for transitions to this other state. So does parity mode evolve faster than climatic tolerance, for example? So we would have some sort of way of modeling transitions between our states. So here we have viviparous and oviparous, and these large arrows represent very fast transitions. Maybe in the tree, we see a lot of changes. And then in climatic tolerance, we have warm climate, cold climate, and maybe the transitions are a lot slower. So that answers a different type of question. Now, they're all related to one another, these questions, but they are a different question. So does parity mode depend on climatic tolerance? Well, we might expect that the transition rates in a cold environment are much faster from oviparous to viviparous than in the warm climate. And this is a real observed pattern that we do see. So this is now a question of dependence. So does my transition from oviparous to viviparous depend on the background climatic conditions? And so to answer all of these questions, we're going to need some data. And in this case, it's going to be data on parity mode and climatic tolerance. So we're going to go to each of these tips, each species. We're going to do natural history work. We're going to dig through libraries of documentations, monographs, all sorts of different things. And we're going to collect data on as many species as we can within our phylogeny. Now, the phylogeny is generated, I should mention this, it's generated separately. So this is taken as a given. So we use sequence data, we construct the phylogeny, we know the relationships beforehand. So we're not going to be inferring the phylogeny in this talk. We're going to take it as given. So now I've gone through, I've collected a bunch of data, and each trip has been scored. So there are two sort of important things to note in my line of research that might differ from how you may apply these sorts of methods. So the first is that I'm using primarily extant data only. So fossil record is a lot better than what people normally think of it, but it's not very good. Darwin described it as a book, and you're missing, 90% of the pages, and of those pages, you have a paragraph, and of that paragraph, you have a few words, right? So the fossil record is pretty scant. It's incredibly important, but for a lot of lineages and for a lot of the characteristics that we're interested in, we might not be able to get data. So climatic tolerance is actually a pretty good example of like the fossil record may or may not be able to give us that kind of information. Depends how well we're able to reconstruct the geology. So I'm going to be relying on extant, which means present day species only. And our temporal information, and so that means we don't know what's going on deep in time. And our temporal information, which I've highlighted here at the very, very far left of the slide, comes from the branch lengths of the phylogeny. So the branch lengths are originally in sort of molecular sequence units, and then you can convert that into a time-calibrated phylogeny with the fossil record and some sort of molecular clock. So our information is going to come from branch lengths and the species positions relative to one another and the distribution of states relative to one another. So now let's answer the questions that we were asking previously. So to do this, we're going to need a model, and this model is the continuous time discrete k-state Markov process, or MK for short. So we're going to have k-states. So in our previous example, we had 4 states, right? Well, we had more, but we had 4 states. We had herbaceous, we had viviparous and oviparous, and we had climatic tolerance, cold and warm. So if we go along this continuous time Markov process, we're going to have our 2 states. In this case, we're looking at herbaceous. This is what the H stands for. So this is like a dandelion. And then we have woody, which is like a tree, right? So this is the life history of the species. And as you go along time, there is some transitions occurring through lineages. So here we're watching a single lineage of plants. It evolves from a herbaceous things. It gains this woody character that we've discretized, and then it jumps back. And all this is happening through time. On a phylogeny, it might look like this. So we have our extant data here. And if we were to paint the branches, if we knew the exact history, it might look like this. So the ancestor was woody, and then at different points, there were transitions to this herbaceous lifestyle. And we represent transitions in sort of this ball and stick diagram where you have some transition rate to W and some transition rate back to H. And this is a memoryless process, which means that your current state is all that matters for your probabilities of transitioning. It doesn't matter how you got to your particular herbaceousness or woodiness. All that matters is that you're the state that you are in now. It's like a game of chess. It doesn't matter. There's a million different ways to get to this position, but once you're in this position, this is what you are dealing with. Okay, so that's what's meant by the memoryless process in our context. Okay, so let's go back to this model. We represent this ball and stick diagram with this Q matrix. So this instantaneous rate matrix is what it's called. And you read this as from the rows to the columns. So if I'm interested in a transition from herbaceous to woody, that is represented by this from row to column. So that's this QHW entry, small QHW subscript. And you also have this woody to herbaceous transition rate. So this ball and stick diagram, this whole process that we are modeling can really be boiled down to the parameter estimates of this Q matrix. And interpreting these parameters is going to be important later on. So Q has units. So these small Qs, they have units. in my field, they're often transitions per millions of years. So we have some transition rate from herbaceous to woody over some amount of time, or per unit time, and my unit of time is usually millions of years. But it depends on the branch lengths of the phylogeny. The units of your phylogeny give you the units of your transition rates. So if we know what the model is, let's make that assumption. So we knew what the parameters of the Q model are. We can now answer, how does X evolve faster than Y? Well, how fast are the transitions in X and how fast are they in Y? Did X evolve before Y? Well, we use the model to predict the unknown ancestral states, and we can now answer the way that evolution has progressed. Does change in X depend on Y? Well, now we're going to talk about this model in a little bit, but we can model X and Y together and check whether X changes more than when Y is in a particular state. So we'll talk about these models in a little bit. So that's the basic picture. You have a bunch of states at the tips and a bunch of unknown internal nodes. Now I want to do just a quick look at parameter inference for this model for anyone who's interested in sort of more of the technical, gory details. So I'm going to randomly pick a set of rates, and we're going to assess the probability of the observed data under these parameters. So we have this rate matrix, right, that we've been talking about, and we have branch lengths from our phylogeny. We can get the probability of a change, so that Q matrix that we were dealing with was in units of transitions per million years. So it's not a probability, right? Well, we're going to need to change this to a probability. So we do that by a matrix exponentiation operation multiplied by the amount of time. So this gives us the probability of changing from one state to another given a certain amount of time. So you can imagine if time is really small, then these probabilities, the probability of going 0 to 0, which is staying in your same state, or the probability of going 1 to 1, those are going to be really high. Okay, so just to give you a feeling for what this matrix is. So as you increase t, you are going to adjust these probabilities. And obviously, the transition rates are also going to be important. So if you have a really high transition rate from 0 to 1, this probability is probably going to be relatively high given any reasonable amount of time. Okay, so we've converted our transition rate matrix into this probability matrix. How are we going to then do our modeling? Well, we take EQT and we multiply it by this probability vector, by this vector over here, which is just the representation now in a numerical form of our tip state. So if this was woody and herbaceous, and this lineage is herbaceous, we'll say red is herbaceous, then the probability is going to be 1 and 0. So that I multiply these together, you can see 1, 0 over there, and that gives me a new probability that I've rescaled for our purposes at this particular branch. Okay, you can repeat that for the other side, and we'll just ignore these two here. And let's say that we get this probability at this particular branch of being 0.2 and 0.8. We can combine those probabilities, and that gives us a probability for this particular node. And we can repeat that for every given internal set of nodes. So I'm going to say that the probability, I want to assess the probability that this was red, this was red, this was blue, this was blue, repeated for all possibilities. And if we were to sum those, we would get the overall probability of the tips. Now, thankfully, we don't have to do that. That would be incredibly computationally expensive. So we do this thing called the pruning algorithm. It's like a dynamic programming algorithm. But essentially, you start at the tips, you work backwards. At each branch, you transition rates over that branch length to calculate the probability, sort of how I was going over previously. You continue this until you arrive at the root. You multiply that by a prior probability, and you get the overall likelihood. I'm not going to go into detail on this, but if you're interested, I'm happy to provide more details and papers that you could read through. So the point is that we're not actually calculating every possible sequence. We are calculating, we're integrating over all of them as we do our pass towards the root. Okay. So we don't have the best model yet. If we've done that for one transition rate matrix, we have to do that over many transition rate matrix. We evaluate the likelihood of any given set of parameters, and then we slowly improve the likelihood until we find our best model. And that's what we go with. But we're not done. Even when you have optimized the parameters there are a lot of different model structures that you can use. So this is our familiar Q matrix once again, but now I'm going to indicate which parameters are being estimated and which are not. So we've been sort of talking about this Q01, Q10. We have a transition from 0 to 1, a transition from 1 to 0, and because these numbers are different, we're saying that the rates are different. Okay, you can imagine this, I don't love the analogy, but you can imagine this sort of being like a selective pressure. If this rate is higher, then the state 1 is being selected for relative to state 0. You can also have equal rates. You can think of this as drift, where you go 0 and 1 and you sort of bounce back and forth with an equal probability of any transition occurring given some amount of time. You can have ordered models. So in this case, you're only allowed to move forward. You only accumulate changes in this case. You never reverse back to a previous state. In fact, I gave a similar talk in Norway to people who study cancer, and they had called these things accumulation models because they were interested in the mutations that accumulate that lead to a cancerous cell type, I guess. I don't know much about cancer. I'm a comparative biologist. So you can have these accumulations where you go from 0 to 1, and that's all that's allowed. You never reverse back, and you can obviously have also 1 to 0. So how we're able to fit these models, we can structure our models such that these are all viable options, and we can fit using the techniques previously, calculate the probabilities, try many different parameters, we can get a likelihood. So how are we going to evaluate these different models? So we want to compare all of these alternatives, and we have ARD, ER, these are all the names of those different model structures that I had shown you. Each has a likelihood. And for those who don't know, if you have a likelihood, the more parameters you have, the better the probability that that model is going to fit your data. It's just the nature of overfitting of your particular data set. If you have a set of points and you draw a line through all of those points, you can fit your data perfectly, but it's way too complex. So we use AIC, which is the Ecology information criterion, and it penalizes our parameterization, saying as you add complexity, you need to improve. You can add complexity only if the improvement is substantial enough. Otherwise, you don't need the extra complexity, and we are trading bias. We are not doing a good bias variance trade-off. Not a stats course. I'm not going to talk about that more. So just as a little analogy, imagine you're packing for a trip. If you're using likelihood, you're going to pack every possible thing that you could possibly need on that trip, and you're going to bring way too much luggage, way too much stuff. If you're using AIC, you're going to pack very organized. You're going to know what you're going for, and you're going to have like a nice trade-off between how much you need to bring and how much you're going to actually use. Okay, is that clear? Good. So for a single trait, 4 models, not bad, right? But complexity can get really out of hand pretty quick. So this was the model that we have been dealing with, just a single transition rate. Now let's add a second binary character. So this was that climatic change with oviparity and viviparity. That's only 2 binary characters. That's not that much information for any given species. But already you can see we have a lot more parameters that are possible, a lot more parameterizations that are possible. We have 8 parameters that are possible here versus 2. Now let's add a third binary character. Again, not too much additional information about the species, but we have exponentially increased the amount of parameters that we need for our model. And that's because every time you add a new state, in theory, you have to consider how that state affects the transitions of your previously included character states. And the thing is, you can run default models, but as you add more characters and more structures become available, you should be considering the alternatives. And I'll show you why in a little bit. But for now, I want to talk properly about these correlated character evolution models. I'm not going to talk about the hidden Markov model yet. That'll come soon. But I want to give you an idea of how we structure these Q matrices such that we can estimate correlated character evolution, or more accurately, dependent character evolution. You want to be like really technical about it. So things are a bit more complex when you study correlated models. So I'm going to give another flower example and say I'm studying the evolution of flower scent. Okay. So we have these two states here, the flower has a smell or the flower doesn't have a smell. And let's say that we know that flower scent should be correlated, or we think we have a hypothesis that it's correlated with white and yellow flowers. And this is true. If you know about floral color, if you are a nighttime flower, you're less likely to have these sort of fancy colors, and you're more likely to attract pollinators through your scent rather than through color. But let's just say we're testing this hypothesis. So we have our two characters, scent and color. Each has two possible states, which I misspelled, which is why this is a different font. So we have scent and unscented, and we have white flower and yellow flower. Let's design our Q matrix considering these four different possibilities. So we're going to read it from row to column. We have scent and white, scent and yellow, unscented and white, unscented and yellow. We don't allow for dual transitions, which means you can't change from a scented and white flower to an unscented and yellow flower. So we've removed all that diagonal from the matrix because those represent the dual transitions. And then we'll just fill it out. And that's actually a dependent model. Now, I don't expect this to click right away because why would it? So let's just focus on a particular transition. Let's say we were interested anyway in this transition from scented to unscented. So let's just look at that. In this case, we are interested in any transition where we go from a scented state to an unscented state. So if we look at this, we're going from these 1 and 2, so that's our scented state. I'll use this so that we're going from 1 and 2, that's our scented state, to 3 and 4, that's our unscented state. So those true transition rates, this 2 and 4 here, that represents just scented to unscented. So if we look at just this parameter 2 from those two that represented the scented to unscented, we are saying, what is the transition rate when scented to unscented occurs with a white flower, right? So this is this transition rate from 1 to 3. So scented plus white to unscented plus white. So given I have a white flower, what was the probability or what was the rate of transition? You can do the same thing. Q24 scented to unscented when the flower is yellow. And you can compare that model where these maybe are freely estimated to a model where they are the same, i.e. they don't depend on the flower color. Regardless of your flower color, you will have the same transition rate from scented to unscented. So you could do you could fit these two models, do a likelihood ratio test, you could use AIC, and you would get evidence or support for a hypothesis of a dependent relationship between these two characters. And that's why, this is really a model of dependence rather than correlation, but we don't need to get into that. So we can gain insights by comparing these different model structures. Okay. And if there is, so using these more complex models of correlated character evolution, because you could, in theory, if we just go back here, I could model scented flowers and white flowers completely separately, right? I could have that binary character matrix, and I only have the 4 parameters maximum. I just separate them out and I model them independently of one another without worrying that the transition from scented to unscented makes, it depends on the flower color. But there are consequences to ignoring this. So this is now a relatively old paper, but was very splashy at the time. It was the ancestral flower of all angiosperms. So angiosperms are the most successful group of plants around. If you look outside and you see something green, there is a really high probability that is an angiosperm. So these are the flowering plants. They all have flowers. That's the character that unites them. And because of this, people are very interested in the different characters of the flora, the flower. So each of these characters in this previous study was treated completely independently of one another, whether I have a little diagram here. So whether your petal changes to a particular orientation, or your stamen, or your sepal, or your pistil, all of these things were allowed to evolve independently. But you can imagine a flower is really an integrated unit. You should be considering how these changes depend on one another. And the character that they were interested in particular, was this whorl versus spiral character. There's a lot of really interesting developmental biology that has been done And in fact, these authors replied to the original paper saying that developmentally, it doesn't make any sense the result you've got. You modeled each of these things independently, you got this weird result. It shouldn't be this mix of whorled and spiral parts, right? And we have really good developmental biology done for flower evolution. So what happens when you use these correlated models? Well, you get something that was predicted by the developmental biologists. So it completely changes your ancestral state reconstruction. And here, what we're looking at is a phylogeny. We don't show the tip data, we just show the big groups of flowers. We won't go too into detail with this, but these are all the predicted ancestral state values. Kind of hard to see with the colors here. That's okay. Okay, so there are some issues with correlated character evolution. I might just skip through this very quickly because I want to make sure I have time for everything. But the quick and dirty thing is if you use these correlated character evolutions, you can have these massive biases towards supporting correlation when there is none. So here we have support for correlation when there shouldn't be any. And so you get a lot of support for correlation. However, if we use a better null model, which is going to be a hidden Markov model, which is the next topic, we can get a much more appropriate support value. And this relies on a hidden Markov model, which again, we're going to be adding some more complexity. And that's what we're going to talk about now. So, hidden Markov models. Well, maybe should I pause for questions? Is that okay? Or yeah, just because of the nature of this. Are there questions before I go on? Yeah.
Sorry, this is more a question about the initial states that you're focusing on. What did you use to determine if a species was warm climate or cold climate? Like,
yeah, so that depends on the character. You can set up some binary thresholds. It's entirely up to the researcher. There's no there's no standard way to do it, I'm afraid. If I was going to do that, I would probably gather data from the location of the species, like their centroid of their range, look at the mean annual precipitate or mean annual temperature, and then do some sort of clustering algorithm. That was a hypothetical study, so I didn't actually do it, but if I was going to, I'd probably do something like that. But it's a really important question, actually. Okay. Cool. So let's talk about hidden Markov models. I'm going to give you another new example. Here we are interested in transitions between a terrestrial habit and an aquatic habit for anything that has a plastid. So this is the group called Bioinformatics. It includes everything from an oak to a cyanobacteria, which are sort of the source of the plastid. So maybe it's cheating, but we are going to model their evolution of their habitat. It's an interesting question to consider. Perhaps where did primary endosymbiosis first occur? Was it an aquatic habitat? Was it a marine habitat? Was it a terrestrial habitat? Under what sort of circumstances would this have happened? Okay, so here's the Viridiplantae phylogeny. We have a bunch of cyanobacteria. We have gymnosperms, and if we were to model this with our sort of standard setup, we are looking at, well, I would expect cyanobacteria to have a much higher transition rate between aquatic and terrestrial than gymnosperms, which are big woody trees. Excuse me. So gymnosperms have a slow transition rate, and cyanobacteria have a fast transition rate. But if we were to model this using our sort of standard setup, we don't have any way to incorporate this great heterogeneity. So that the fact that some lineages evolve fast and some evolve slow, in our standard setup, there is no good way to include that information unless you're using a dependent model. So that's where these hidden Markov models come in. We define things in terms of different rate classes. So you have aquatic to terrestrial in rate class A, and you have rate class B. And rate class A is a slow rate of evolution, and rate class B is a fast rate of evolution. And lineages can transition between these sort of meta states, these rate classes, freely. So let's go back to our Q matrix. We have transitions from A to T and T to A, so aquatic to terrestrial and terrestrial to aquatic. That's occurring here. But we also now have this rate class 2, which has aquatic to terrestrial and terrestrial to aquatic. Good. So we've now represented this and this, all that's left to do is to add in transitions between the rate classes. So we have transition between R1 and R2 on this off diagonal, and we have transitions between R2 and R1. So now we have our full model, and it's really that simple. We use the same machinery, and now we have created our hidden Markov model. And what's happening here is we sort of have a process that controls another process. So we have fast evolution, we have slow evolution, then we have a Markov model switching these two states. But all that we've observed is the aquatic and terrestrial. We are going to infer these rate class assignments based off of our observed data. So the fact that there's very few transitions over here and very many transitions over here. I'll have a figure about that in a little bit. So where is our information coming from? It's coming from the differences in the transition rates. So here we see like these sort of yellow areas. This is my fault. I should have had a higher quality figure. It's been degraded through time. But you have like areas of the phylogeny where you have very high transition rates, and you have areas of the phylogeny where you have very low transition rates. And that's where the information for the hidden Markov model is coming, even though we never observed directly rate class 1, rate class 2. It's all inferred from the extant data. Okay, there's another way to think about HMMs in comparative methods, and this was popularized originally, actually. So when we first started using this in my field, it came out as a precursor model. And I should say, hidden Markov models have their origin in molecular evolution, something called the covariant model, if anyone's familiar with it. So it's this idea that DNA evolution is either happening and occurring in one rate class, or it's completely off and never transitioning. And so you can transition between these two things. And that's like the true origin of hidden Markov models within sort of phylogenetics as a whole. This is when they were first applied to comparative biology. So we have this system of guppies, we're going to say. And what I observe is male coloration. I know if the male is plain or if it's very ornate. And so I might go to Trinidad and collect a bunch of guppies and look at all the different colorations that are possible. And I just say, is it plain or is it ornate? And then we can do our modeling, right? The thing is, the transition from plain to ornate is probably going to be modulated by this unobserved character, in the sense that I can't literally measure it with like a camera, of female preference. So before we get sort of selective pressure to see an ornate male, females have to prefer that phenotype. And so there's this unobserved state, this hidden state is what we call them. So the way that we would model the evolution here is that we expect that maybe the lineage starts in a plain fish, a plain male fish, and females don't have a preference. Then this hidden state develops where females do have a preference, and then we get the evolution of the red coloration, of the ornate coloration. And this is how you would represent that model in matrix form. We don't have to go over it. We've seen a lot of matrices already. Okay, ancestral state reconstruction. It's never ending. Okay, so ancestral state reconstruction. You've probably noticed that in comparative biology, we are interested in reconstructing the sequence of events, how they've occurred. We use these models as sort of the framework, which we're going to build off of to make estimates about the past. So this could let us say what the derived state is, which is ancestral. And in a lot of studies, actually, the model parameters that I've been talking about, these model comparisons, they're really just nuisances before you get to your ancestral state reconstruction. You need to have an optimized model before you do your historic sort of inferences. So how do we estimate unknown states? Well, we have the marginal probability. There's two main types that are used. So the marginal probability asks what's the probability of a particular state considering all other possible states and all other nodes. And you represent it by this pie chart. So here, the probability of a state 1, 0 is very high, and the probability of a state 0, 0 is kind of low, and then 0, 1, and 1, 1 aren't even represented at all. But the way you've got this probability is to say fix this state in maybe 0, 0 or in 1, 0. We'll start with 0, 0. You fix that in 0, 0, and you evaluate the likelihood of everything else, integrating over every possible different scenario. Then you do the same thing for 1, 0, do the same thing for 0, 1, and you do the same thing for 1, 1. you rescale them so that they sum to 1, and you get these pie charts. So you're considering all possible scenarios. Now, should I go into the technical details? No, I'll give an example after. Okay, so then the joint is different. This is now with a hidden Markov model. But under the joint, we are interested in considering the most likely set of states for a given observation. So here we have our model, and you can see that these pie graphs are now completely filled in, and that's because this is the most likely sequence that led to our observation. So the example that I came up with that I don't have a figure for it is, imagine that you are driving through a set of 3 traffic lights, right? You can get any order of these traffic lights being red and green. You could have red, 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 you can have green, 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 whatever it is, right? And let's say that there is some sort of conditional dependence. So if you get a red light first, you end up getting reds all the way through. So that's one very likely scenario. And the other scenario, you get green first and you get greens all the way through, right? So the joint reconstruction would, unfortunately, just by the way that things are programmatically, would just give you one of those 2 scenarios, or give you the most likely of those 2 scenarios. Now it's ignoring all the other possibilities of red, green, 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 red, red, all of that. But it would give you the probability of each different scenario. It's just the algorithms we use only return the best one, although we're working on that. But that's what it would give you. It would give you red, red, green, or red, red, red. The marginal considers every possible scenario and then summarizes them. So the fact that red, 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 and green, green, green are equally likely means a marginal probability of red, red, and green is 50-50 at each of the possibilities, at each of the traffic lights. So it's somewhat misleading. Now, it is accurate in its own way. It's just not telling you maybe the most important thing. But that's the difference between marginal and joint. So another big issue in the field I was talking about is these different model structures. I'll skip over those. Okay, so let's get to the example. Actually, ahead of time. So that's good. So this is in collaboration with a bunch of people through Midge. Kyle's here. Anyone else here? I don't know. They're not creative, basically. And we're interested in, and honestly, Kyle can probably tell you more about the system than I can. I'm just going to tell you how we're using it. We're interested in antibiotic resistance in different bacteria. And I think the bacteria that we're using is Klebsiella.
Yep.
Yeah. So it's been on the rise, it's an urgent threat, and I'm going to show you how we've been applying these models. Yeah, so here is the timeline for the resistance emergence. Okay, so we're gonna start with the phylogeny. Here's our phylogeny. We're gonna need state data. So in this case, maybe we're interested in AMK, which is amikacin resistance. We have susceptible individuals in gray. We have resistant individuals in black. Maybe we're interested in CST, which is colistin resistance. Again, susceptible in gray, resistant in black. Or maybe it's IR and Miro, which are
imipenem, that's a beta-lactamase inhibitor, and meropenem is carbapenem, so a beta-lactam antibiotic.
So those things, I'm not repeating that. So we can put all of these different things at the tips of our phylogeny. This is the resistance that we're interested in seeing, like, well, when do they gain resistance? When do they lose resistance? When do these different lineages have these evolutionary transitions? So maybe if we're looking at just one of them, we can use these familiar models, right, that we were talking about. We can look at the model of susceptible and resistance on the phylogeny as changing between susceptible and resistance. So maybe this is the gain of resistance, and we say that it might be faster or slower than the gain of susceptible. Maybe we say that they only gain resistance, or that they have an equal rate, equal probability of gaining it or losing it in any step in time. And we compare these models. We can also, if we go back to that IR Miro example, we might say that there is a correlation between them, right? We might say that if you gain resistance in IR, you are more likely to gain resistance in a different antibacterial, right? That's another hypothesis that we can compare. What we were doing was actually using these hidden Markov models, and we're not done with this. The story doesn't have like a super interesting conclusion, I'm afraid, but this is the setup. We were using these hidden Markov models where we are interested in a particular resistance type, so maybe it's IR or Miro, and we think that there are differences within the lineages in how they gain and lose the resistance. So we applied this to a bunch of different antibiotics. And it's hard to see here, so let's just zoom in on one of them. And what we're looking for are places where you move from this slow rate class, which are these lighter colors, to places where you have this high rate class. And this high rate class represents a shift in how the lineages are undergoing resistance evolution, essentially. Where are they starting to go maybe more quickly gain resistance, or maybe where they're more likely to lose resistance. And we can identify different nodes in the phylogeny. So maybe this is where those transitions are occurring. Your mesh is up exactly where I want it. That's okay. And we can apply then, combining this information, which we can get using ancestral state reconstruction, we can then do an ancestral sequence reconstruction. So we can get the sequences of the extant lineages, reconstruct their sequences to the past, correlate it with where we expect these transitions are occurring, and then look at the sequences themselves and try to highlight different parts of the genome that we think are associated with these transitions between susceptible and resistance. And then the next step is like doing the actual sort of scientific work and testing our hypotheses. But that's where we are now. And that's it. That's all I have for you today. So I would take questions.
I have a question on the joint ancestral state reconstruction. You say that it provides the best possible outcome ultimately. Do algorithms also provide the second best, third best, fourth best? Is that parsable or able to find? Is that not very common?
Not until what Stephen and I are doing now.
Okay.
So we're like we have an algorithm now that will give you the sort of confidence intervals around any given ancestral or ancestral estimation.
Okay.
Yeah, because otherwise it's a problem. And the thing is, like, these joint reconstructions actually work a lot better than the marginal. So, you know, the obvious thing to do, and this is what most people do, is if you have these marginal probabilities, you just take the maximum a posteriori estimate. So whichever is the largest section of pie chart, that's what you're going to use for your ancestral sequence, right? So if we look at the traffic light example, you are essentially just going to each intersection saying, okay, 5149, 5149, 4951. You can get any number of different things, right? So it's not the best practice, and in fact, it doesn't work very well. So it's about 3 times less likely to give you the true ancestral state reconstruction than if you were to use this joint. It's because there are sort of these conditional dependencies they exist in the marginal probability, just not in an intuitive way. So it's easy to misinterpret. The marginal probability is fine. Like, there's nothing wrong with using a marginal. It's just the way we interpret things tends to be tricky. Sure. Yeah.
With a lot of these models, are there like only one defined sort of like attractor to like all these like states, or can there be like multiple valid like hypotheses that can come from running these like models?
What do you mean by the attractors?
Well, it's like say there was like a stable state that was met with the evolution of like cyanobacteria and woody plants. And then that only, like it's stable and it converges to really only one configuration between them, but could it be like multiple?
Yeah, so you could do that. It would be up to you to structure the model in that way. And it would be valid to test those different hypotheses. Obviously, as you add more complexity to the model and you want to look for maybe these alternative sync states or stable states, you are going to run into data challenges because you have a lot of parameters that you're going to need to fit, but it is in theory possible. And in some work that I was doing, I was actually looking at automatically trying to infer these different model structures. And it does make a massive difference. So exploring these different model states or these model structures is massively important. It impacts everything from your rate estimates to your ancestral state reconstructions. So it's a really good thing to think about.
Any questions?
Actually, I have another
Yeah,
follow-up. So would it be a better advantage to look at like when we were talking about like looking for configurations that like maximize some sort of likelihood? Would it then be like a good one if you're returning multiple parameters, like we were talking about, like the 5 best models? Would that just be like the 5 best models with, you know, the best possible likelihood or AIC? Or would that be like the best possible within like certain groups of like likelihood peaks or something like that?
Yeah, it's probably representing the best possible of likelihood peaks, if I'm being honest. They're hard to optimize these models. They can get pretty tricky pretty quickly. Yeah, you define sort of the space that you're willing to search. Generally, people will use just the best model. So a single sort of like a point estimate of the model structure, and that model structure is going to have estimates obviously for its transition rate matrix. It's tough to search like the full potential model space, I'm afraid. There are some interesting ways that these models group together, like Lie Markov groups was something I was interested in. I couldn't figure out how to sort of apply it, but there are like natural groupings within these models that could make searching them a bit easier, and maybe like graph like embeddings of the model structures could be useful. But yeah, right now the way things are, it's like you set up the model you want to compare, use AIC. That's where you get.
Cool.
This is more of a question on speciation or new application of these models to, I don't know, predict or identify possible speciation events. How would?
Yeah, so that's actually another class of sort of these models that builds off of it. So what we had been discussing, I just want to find I'll just go to an early ball and stick diagram. This will be fun. So we've been discussing just sort of transition rates between observed phenotypes. What you can also do is use a birth death process where you have some sort of speciation rate and some sort of extinction rate and associate it with these characters. So you say if it has a resistance, right? Maybe it has a higher speciation rate and a higher extinction rate. If it has, so say if it's susceptible, it has a lower speciation and a lower extinction rate. These are called state-dependent speciation extinction models. And I mean, that's how they work, right? If you have this red character, you are more likely to have a speciation event occur or an extinction event occur. If you have a blue character, you're maybe less likely. Your rates are lower when you have this blue character. So it combines these Markov models with a birth-death process. So it's a very, it was like a very hot area of research for a long time, still is, but there are problems with our data because I'm using only extant with the types of data that are common in bacteria or viruses. You have heterochronous sampling, so you might be able to infer this a little bit better. Yeah, very cool set of models. Does that answer